Good morning. Hope you enjoyed this session about the karyotype. We will be working together in Activity 1 of Chapter 2. You will find it in Unit 1 of your textbook. So, what is a karyotype? What does this term represent? A karyotype is a photomicrograph of the complete set of chromosomes of a cell arranged according to specific criteria. So, as you can see in this figure, the chromosomes within a cell are not arranged. That's why we are going to take a photo of these chromosomes and then we are going to arrange them according to specific criteria that we will discuss later on. This photo of arranged chromosomes allow us to study these chromosomes to detect any chromosomal abnormalities. Let me remind you about chromosomes and where they are located within a cell. In this following video, genetic material is the hereditary material consisting of chromosomes that carry the information for determining the characteristics, or traits, of an organism. The following short video will show you that chromosomes are located within the nucleus of a cell. Before going into the details of karyotyping, and the different steps involved in preparing a karyotype, we are going to take a look at the different types of cells within our body. Our cells can be divided into two main groups. Somatic cells, or also known as the body cells. These cells are produced by mitosis, and are not involved in reproduction. For example, skin cells, or muscle cells. Somatic cells contain diploid number of chromosomes, which means that each chromosome is found in two copies within these cells. The other type of cells are the sex cells, also known as gametes. These are the sperm cells in males and the oocytes in females, and for sure you remember that a sperm can fertilize an egg resulting in a zygote, which will grow into a new whole organism. Gametes are haploid cells, containing haploid number of chromosomes, meaning that each chromosome is found in only one copy. Sex cells used for sexual reproduction are produced by meiosis, during which the original diploid mother cell divides and its genetic material is reduced by half, resulting in haploid gametes. To better understand and know the difference between the terms, somatic cell and gamete, as well as, diploid cell and haploid cell, please watch this video.
Alright, so in this quick video, we're going to go over the basic differences between haploid cells and diploid cells. Well, before we get into haploid and diploid, we kind of have to put down some ground rules as far as some terminology. So, let's look at what are called somatic cells. And like it says, somatic cells are defined as body cells, cells of your body. Non-sex cells. So examples of cells of your body: skin cells, brain cells, nerve, muscle, white blood cells, red blood cells. And so these are what we mean by somatic cells. What we're going to do is we're going to zoom into this woman's shoulder right here. We're going to zoom on in and take a look at skin cells as an example. Let's go ahead and do that right now. And so when we zoom on in, we can see a layer of skin. Well, let's zoom on in even closer. Let's zoom on in at one particular cell, one particular skin cell. And so now we're looking at one particular skin cell. But you know what? Let's zoom on in even further. I just highlighted a nucleus of a skin cell. Let's zoom on into that nucleus, and let's see what's inside the nucleus. And when we look inside of the nucleus of cells, whether it's a nerve cell, a skin cell, a brain cell, when we look inside the nucleus, we find chromosomes. And that's what these stained rainbow-shaped structures are. These are chromosomes. Now, when you arrange chromosomes into an organized picture, you have what's called a karyotype. Now we're getting into what's a diploid cell. Notice how these chromosomes are arranged. There's two chromosome number 1s, and two chromosome 3s, and two chromosome 10s, and two chromosome 18s. Diploid cells what the word diploid means is that they are cells where their chromosomes are paired. Notice how there's two chromosome 6s. The reason there's two chromosome 6s is we inherit one from each parent. The reason there's two chromosome 4s, we inherit one chromosome 4 from our mother, one chromosome 4 from our father. So somatic body cells are also diploid. Diploid means that they have paired chromosomes. And in humans there are a total of 23 pairs of chromosomes for a total of 46 chromosomes. 23 pairs adds up to 46 total chromosomes. Diploid cells are also created by the cellular division process known as mitosis. Well, again, before we go into haploid cells, we need to examine some other terms first. We need to examine what are called gametes. Gametes, by definition, are what we call sex cells. Now, we're going to look at a woman in this example, but keep in mind that men also have sex cells. So in our picture here, we have a woman's reproductive system, and what we're going to do is we're going to focus on her ovaries in just a moment. So gametes, sperm cells and egg cells. But let's zoom on in. We're going to zoom on in this example to female gametes. But men, we also have gametes. Our sperm cells are housed inside of our testes. We're going to let's zoom on into a female ovary right now. And when we zoom on into a female ovary, we see a bunch of cells, a bunch of egg cells inside the ovaries. Well, let's zoom on into one single egg cell. And so here we have one single egg cell. And just like the last time, there's a nucleus to that egg cell. Let's zoom on in and take a look at the nucleus of the egg cell. So far everything pretty much looks the same. Inside of the nucleus of an egg cell, also inside the nucleus of a sperm cell, but inside the nucleus of an egg cell we have a bunch of chromosomes. But watch this, here's the difference. When the chromosomes are arranged from biggest to smallest, chromosome number 1 is biggest, number 2 is a little smaller, number 3 is a little smaller. I hope you see the difference. Now we're coming to what haploid cells are. Haploid cells are cells such as the sperm and egg. Haploid cells are gametes. Haploid cells are cells with a single chromosome. There's only one chromosome number 1. There's only one chromosome 15. There's only one chromosome 17. So I hope you see the difference. The chromosomes are not paired as they were in the previous example. Haploid cells, if we look at humans have a haploid chromosome number of 23. That means in a female egg cell, there's only 23 chromosomes. In a male sperm cell, there's only 23 chromosomes. Remember, that's kind of different from what we said a moment ago. If cells are diploid in humans, if they're diploid, they're going to have a total of 46 chromosomes. But cells that are haploid only have half of that amount, 23 chromosomes. And so these cells are created by meiosis, the cellular division process known as meiosis. Finally, when we close out this video, let's put the karyotypes, two karyotypes of a diploid cell on the left and a haploid cell on the right, and just put up some of the main differences between the two. So here is just a nice picture summary of the differences between diploid cells on the left and haploid cells on the right. I hope this video was helpful. Go ahead and pause the video if you want to examine this picture in a little more detail.
All nucleated cells contain chromosomes within their nucleus. And these chromosomes are classified into two types, autosomes and gonosomes. So what do we mean by these terms? Autosomes or autosomal chromosomes are chromosomes that carry genetic information responsible for general body characteristics of both males and females. While gonosomes or gonosomal chromosomes, which are the X and Y chromosomes, are mainly responsible for sex determination, such that each diploid cell of a female contains, in addition to the autosomes, two X chromosomes. while the diploid cells of a male contain one X chromosome and one Y chromosome. In the next video, you will be better introduced into this chromosomal classification. Let's watch it. Now,
this is a much simpler type of classification. Here, the chromosomes are classified whether they are autosomes or sex chromosomes. Now, what could these be? This is a representative image of a typical human karyotype. The chromosomes that pair from 1 to 22nd are called autosomes. That is, they are not the sex chromosomes. To be precise, they have got no function to deal with the sex of the individual. The last pair, however, determines the sex of the person. So if the two are identical, that is, both are X and X, then the individual is a female. And what if both are different? In such a case, when one chromosome is X and the other is Y, the individual is a male. This pair is the sex chromosomes. So the chromosomes which help in determining the sex of an individual are sex chromosomes, or even called allosomes. The others, which are from 1st to the 22nd pair, are not the sex chromosomes. This makes them the autosomes. Both contain genes that code for proteins which make up our body.
Now, let us recall some facts about human chromosomes. What is the number of chromosomes found in the nucleus of a human somatic cell? How many autosomes are there? How many gonosomes? What is the chromosomal formula of a female somatic cell? What is the chromosomal formula of a male somatic cell? What is the chromosomal formula of a female gamete? What is the chromosomal formula of a male gamete? Pause the video and take some time to answer. Now, correct your answers. A human cell has a total number of 46 chromosomes. 44 of these 46 chromosomes are autosomes and the remaining 2 are gonosomes. In this example, the gonosomes are X and Y, so this belongs to a male cell, otherwise, it would be XX for a female cell. When we say 46 chromosomes, we also consider them as 23 pairs, so we can say that there are 22 pairs of autosomes, and 1 pair of gonosomes, within each human somatic cell. Now let us look at the chromosomal formulas. The general chromosomal formula of a human somatic cell is 2n equals 46. Letter N representing the number of types of chromosomes. Knowing that, we have 23 different types of chromosomes within the human cell. The number 2 representing the number of copies of each type. Thus, 2n means that there are 2 copies of each chromosome type, that's why somatic cells are diploid cells. So, in other words, 2n is 2 times 23. This is equal to 46, which represents the total number of chromosomes within a human somatic cell. To go into more details. In case of a female somatic cell, we would have 44 autosomes and 2 X chromosomes. Hence we say, 2n is equal to 44 plus XX, or we can also say, 46, XX. Note that, when we use comma, this means that the 2 X chromosomes are included within the total 46. But, in the case of a male somatic cell, which contained equals SXY not XX, the chromosomal formula would be 2n equals 44 plus XY. Or, 2n equals 46, XY. Now, let's think about the sex cells. In the case of gametes, which are haploid cells, the chromosomal formula would be 1n equals 23. This means that, there is only 1 copy of each chromosome type. 1 copy times 23 types is equal to 23. Remember that a human gamete contains a total of 23 chromosomes. 22 of which are autosomes, and only 1 gonosome. So, in the case of a human female gamete, the chromosomal formula would be 1n equals 22 plus X, that is 22 autosomes plus 1 X chromosome. Female gametes always carry an X chromosome, never a Y chromosome. While in the case of a human male gamete, the chromosomal formula would be either 1N equals 22 plus X, or 22 plus Y. There are two possibilities because males have one X and one Y chromosomes, and therefore, the sperm might carry either X or Y chromosome in addition to the other 22 autosomes. Chromosome abnormalities are responsible for a significant portion of genetic disease in the population. Have you ever heard of the terms Down syndrome or trisomy 21? How can you detect such abnormality? Do you remember? Chromosomal abnormalities can be detected using karyotyping technique. So what is karyotyping? It is the process of arranging chromosomes based on a certain criteria in order to count them and to detect chromosomal abnormalities. What you have to know is that the karyotype test should be done during cell division. Let us look why. As you can see in this figure, the aspect of the genetic material is different among these two cells. The first cell is not undergoing cell division, and if you look at the genetic material within the nucleus of this non-dividing cell, you will not be able to see clearly the different chromosomes. The genetic material is dispersed throughout the nucleus, and is termed chromatin. While in the other cell which is undergoing division, the chromosomes are clearly seen and we can even count them. In this case, the genetic material of this cell consists of 4 chromosomes. Based on this observation, we can now understand why karyotyping is done during cell division. It is simple logic. The karyotype test is done to allow the detection of any chromosomal abnormality, and so, we should be able to see the chromosomes to count them and examine them. That's why a karyotype test is performed on dividing cells. But before going into more details, let us see the different states and aspects of chromosomes within a cell, before cell division and during cell division. Chromosomes exist in different states and aspects throughout the life of a cell. When the cell is not dividing, that is before cell division, the chromosomes are non-condensed, they look like long loose structures, and they can be either formed of one chromatid, or of 2 chromatids. In this case, the 2 chromatids of a chromosome are held together at the level of the centromere. Of course, when the chromosomes are not condensed, they are not well observed within the nucleus, and the complete set of non-condensed chromosomes within is known as chromatin. On the other hand, when the cell starts division, the chromosomes will condense and they can be clearly seen under the microscope. Of course, at the beginning of cell division each chromosome is formed of 2 chromatids, but later on, the chromosome would have a different aspect. It will be formed of 1 chromatid. As you will see later, when we study mitosis. So, to summarize the main idea, chromosomes exist in different states and can have different aspects depending on the stage of the life cycle of the cell. As you will see in the next activity. A chromosome can be non-condensed of 1 chromatid, non-condensed of 2 chromatids, condensed of 1 chromatid, or condensed of 2 chromatids. And it is during this stage where a karyotype is prepared to be analyzed. This figure shows a chromosome of 2 chromatids attached at the level of the centromere. This is the aspect of a chromosome during cell division, observed in a karyotype. In this slide, we will see the steps followed to prepare a karyotype, so, we are going to describe the procedure as shown. First, blood sample was taken and cells were separated from the serum by centrifugation. The white blood cells were removed and cultured in vitro to stimulate the cells to divide. After that, the mitotic division was stopped at metaphase, and a hypotonic solution was added to swell the cells. Then these cells were spread on a slide, fixed and stained. Finally, the chromosomes were examined. Now, let me explain some main steps. You know that blood contains different components, but since we are interested in studying chromosomes, so that's why we need to work with cells, specifically nucleated cells, because, as you have seen earlier, the chromosomes are found within the nucleus. Blood cells include red blood cells and white blood cells. The red blood cells do not have nucleus, so, we say these cells are enucleated and they are not needed in this case, but the white blood cells are nucleated, having nucleus, so, we are interested in these cells in our work. Moreover, we have said that chromosomes can be only observed when they are in the condensed state, which is during mitosis. Of course you know that by mitosis we mean cell division, and this type of division consists of different stages including metaphase, during which chromosomes are fully condensed and can be observed clearly. That's why we have to stimulate cells to start mitosis, so that chromosomes will condense, and then we stop mitosis at metaphase and fix them on a microscopic slide to examine them. Note that mitosis can be stopped at metaphase by adding a chemical known as colchicine. Now let us see the role of the hypotonic solution. When we say hypotonic solution, we mean a solution that has a concentration lower than the concentration of the fluid found within our cell. According to the law of physics, water will move into the cell and the cell will be filled with water and will swell, that is increase in volume, so that we can better see the chromosomes inside the swollen cell. Here is a video showing you the karyotyping procedure, done in the laboratory. This is how the photograph of the chromosomes looks like during metaphase. Now, we are going to arrange them according to certain criteria as shown in the next slide. After completing the karyotype arrangement, let us look how the arrangement was done, and I mean according to what criteria are the chromosomes arranged. As you can see in this figure, the chromosomes are arranged in pairs, but why do we arrange them in pairs? Of course, because in this case we are working with human white blood cells, and these are diploid cells having 2 copies of each chromosome type. That is 2 copies of each autosome and 2 copies of the gonosome. The chromosomes of the same autosomal pairs are called homologous chromosomes. One is of maternal origin, that is inherited from the mother, and the other chromosome is of paternal origin, that is inherited from the father. Of course human diploid cells contain 22 types of autosomes, and there is 2 copies of each autosome, so, normally, there should be 44 autosomes. Moreover, these cells contain 2 gonosomes, and we can see them in this karyotype. 1X and 1Y. So, in total, there is 46 chromosomes arranged as 23 pairs. The autosomal chromosome pairs are arranged in decreasing order of size, from largest to smallest, and then we put the gonosomes. Now look at each autosomal pair. You can see that the chromosomes have the same size, same centromere position and the same bending pattern. So, when we say homologous chromosomes or chromosomes of the same type, have the same bending pattern. What do we mean by bending pattern? It will be explained in the next slide. Specialized stains or dyes produce unique bending patterns along each chromosome, that is, the chromosome can be stained to be seen in either black and white, or in colors. Bending patterns are extremely helpful for detecting any abnormality in the chromosome structure. See in this figure, the dark and light bands, or the black and white lines along each stained chromosome. And you can notice that different types of chromosomes have different bending patterns. So the bending pattern of chromosome number 1 of a human cell would be different than the bending pattern of chromosome number 2. So, in summary, we have learned that chromosomes are arranged as based on their size, centromere position, bending pattern. And the criteria of arranging chromosomes to obtain the karyotype is as follows. Decreasing order of the size of autosomal chromosomes. Homologous chromosomes are arranged in pairs, such that these chromosome pairs have the same size, the same centromere position and the same bending pattern. This is another figure showing chromosome arrangement, and I want you to know that sometimes the chromosome aspect is not clearly seen. For example, look at this chromosome, It is formed of two chromatids, but the chromatids are not well seen. To avoid any misconception, you have to know that the chromosomes in a karyotype are always condensed and formed of two chromatids because the chromosomes are observed during metaphase. See this video. Hope the video was clear enough.
Human chromosomes can be like this as a single component, or they can be replicated, like this. In our chromosome counting video, we mentioned when chromosomes are duplicated during interphase, the newly made chromosome is still bound here in this region called the centromere. By the way, this region does not technically have to be in the center of the chromosome. Because of it being joined here, we still count this as one chromosome unit. But that can be kind of confusing. We say that there are two sister chromatids. So one chromosome here, it duplicates, and now, while it is still attached, it's still one chromosome. So in humans, you have 46 chromosomes. In interphase, they are duplicated. There are still 46 chromosomes here, but that's 92 chromatids in the picture. During a stage of mitosis, those 92 chromatids get pulled apart, so each cell will have 46 chromosomes. Now, just a side note, while most of your body cells have 46 chromosomes, it's important to remember that certain genes on the chromosome may be turned on or off by a variety of factors. For example, you wouldn't want your eye cells actively using a gene to produce stomach enzymes. That'd be a problem. So, would you ever get a chance to see all 46 of your chromosomes clearly? Well, you could in something called a karyotype, an image of all your chromosomes. It's possible to have a karyotype made as part of some types of medical screenings. In a karyotype, the chromosomes are stained and visible. The chromosomes from a karyotype are typically from a cell that is in a mitosis stage, typically metaphase. As chromosomes are condensed and thick, a karyotype during interphase would be a challenge because you don't have that condensed DNA packing. Now, something confusing to clarify. When searching for karyotypes, sometimes it'll be represented kind of like this diagram, and sometimes you see something like this. And while both show 46 chromosomes, the number of chromatids in the two images here seem different, so what's going on? Now, while the mitosis stage is anaphase or telophase would be a time when each chromosome would have a single chromatid, those phases are generally not when you do a typical karyotype. For optimal viewing, a typical karyotype is taken at or right before metaphase. Recall from mitosis that in metaphase, the chromosome would have two joined sister chromatids. It's just that many times the joined sister chromatids of the chromosome can be so close together that to an untrained eye, it might be difficult to see that each chromosome in the karyotype technically has two sister chromatids. When arranging the karyotype, the chromosomes are arranged in homologous pairs. Homologous chromosomes are about the same size and contain the same types of genes, and in each homologous pair, you receive one chromosome from one parent and one chromosome from the other parent. 23 pairs. 22 of the pairs, 44 of the chromosomes, are called autosomes. That means they are not related to your biological sex. They may have genes related to eye color or height, or hair texture. The last two chromosomes are called sex chromosomes because they determine biological sex. Females have two X chromosomes, males have one X and one Y chromosome. Oh, and just a side note, the name X and the name Y have nothing to do with the shape of them. The reason is an interesting topic to look up, though. So 23 of these chromosomes are from the father, from a sperm cell, which is a gamete. Unlike body cells, gametes have the number of chromosomes as body cells. Sperm cells are considered haploid because they only have one set of chromosomes. The other 23 of the chromosomes came from the mother, contained in an egg, which is also a gamete. An egg cell is haploid because, like the sperm cell, it contains only one set of chromosomes. When a sperm and egg cell combine, they form a fertilized egg, known as a zygote. The resulting cell is diploid, as it contains two sets of chromosomes, and it will divide to continue to form more diploid cells. You are a diploid organism. Now, if you look at the sex chromosomes, you know that mothers can only give an X chromosome, because that's the only type of sex chromosome they have. But fathers could give an X, the baby would be XX, female, or a Y, the baby is XY, male. So fathers determine the biological sex. So why do we care about karyotypes or learning about chromosomes? Well, one reason is it can really help when we are trying to understand genetic disorders. A karyotype can reveal missing chromosomes or extra chromosomes at specific locations. Do you have an interest in a career that involves this area of study? The field of genetics continues to expand, and the career of a genetic counselor may be something to read up on. Well, that's it for the Amoeba Sisters, and we remind you to stay.
Okay then, we have prepared a karyotype, but what is the aim of karyotyping? The karyotype allows us to study the set of chromosomes with respect to number and structure, to detect any chromosomal abnormality. Determine the sex of an organism, if male having XY or female having XX. Determine the characteristic number of chromosome of species, that is how many chromosomes does an organism of a certain species have. For example, humans have 46 chromosomes but chimpanzees have 48. Classify a cell as diploid or haploid. In this case, we have to inspect whether the chromosomes are found in 2 copies or only 1 copy. Now that we know the objective of the karyotype test, which is allowing us to identify chromosomal abnormalities if present, so, let us study the different types of these abnormalities. Chromosome abnormalities could be due to a change in the chromosome number. For the first karyotype, if we count the chromosomes we can see that there is 47 chromosomes instead of 46 chromosomes. This type of abnormality could be due to an extra autosome or an extra gonosome. So, we should inspect each pair at a time. You can notice that all chromosomes exist in 2 copies except for the chromosome number 21, which exists in 3 copies instead of 2 copies. Such type is known as trisomy. Tri means 3, as in 3 copies. The same is done for the second karyotype, but in this case there is gonosomal trisomy, that is, there is 3 copies of the gonosome instead of 2 copies. So, instead of having 2x, or 1x and 1y, there is 2x and 1y. In this karyotype, you can see that there is only 1 gonosome instead of 2. This is also considered as numerical abnormality, but, in this case there is a missing chromosome, so in here, we have monosomy. What you have to know is that chromosomal abnormalities might be related to chromosome structure and not only number, as you will see next. Another type of chromosomal abnormality is a structural chromosomal abnormality, in which the structure of a chromosome is affected. Let us take a close look at the homologous chromosome pairs and compare them. You can detect 2 differences among 2 pairs, pair number 9 and pair number 22. You can also see that in pair number 9, one chromosome is longer than the other, that is having an extra part. While in pair number 22, one chromosome is shorter than the other, so, a part is missing. In this case, a part of chromosome 22 was broken and was translocated onto chromosome 9. Such type of chromosomal abnormality is known as translocation and it is a structural chromosomal abnormality. To summarize, there are 2 types of chromosomal abnormalities: numerical or structural. In case of numerical chromosomal abnormalities, there is variation in the chromosome number. There will be either 1 extra chromosome, and the abnormality is known as trisomy. Or there will be 1 missing chromosome, and the abnormality is known as monosomy. But in the case of structural chromosomal abnormalities, there is variation in the chromosome structure. Hope you have well understood this topic. See you later in the next session.